All right. We're
And this
echoed a
one is
little
good.
bit. Oh.
Good afternoon. Welcome to Medicine Grand Rounds. It's my great pleasure to introduce our speaker today, Dr. Karen Pierce. Dr. Pierce is a professor in the Department of Neurosciences at the University of California, San Diego, and also serves as the co-director of the UCSD Autism Center of Excellence. She has for the past 25 years, studied the autism spectrum disorder, focusing on early detection, eye tracking, and brain imaging. She developed the Get Set Early model, which mobilizes pediatricians to identify autism spectrum disorder around a child's first birthday, enabling rapid access to services. Her work has been funded by a range of agencies, including the NIH, the CDC, the Simons Foundation, and has revealed that early biomarkers of autism can be detected as young as 12 months of age. Dr. Pierce has over 100 publications in top-tier medical journals and is an invited keynote speaker across the world. She has received numerous awards and honors, including an NIH Merit Award, the UCSD Donald Tuzin Community Service Award, and the San Diego Health Hero Award. Today, she will be speaking to us about the clinical and biological heterogeneity of autism and how early eye tracking and brain imaging markers can shape the future of autism research and clinical care. Thank you so much, Dr. Pierce.
And thank you so much for inviting me. I always really enjoy kind of spreading the word about our research and heightening awareness about autism. So thank you so much for the invitation. Okay, so first I have a disclosure statement that I have developed a series of eye tracking tests. They're licensed through UCSD and they're licensed through a company, and I'm a consultant for that company. Okay, so as you mentioned, I'm the co-director of the UCSD Autism Center of Excellence. We do lots of different types of research: early screening, diagnosis, and prevalence. We do genetics research, brain imaging, stem cell, eye tracking, and we have over 20 scientists and staff. We've published collectively over 200 papers on autism in, journals that get wide dissemination, because we really want to make sure that everybody really understands what's going on with autism. So the thing about autism, if you're an autism researcher, that's incredibly interesting and important, is that there's considerable heterogeneity. Consider we have a person named Stephen Wiltshire. He has autism. He didn't speak until he was 7, but he's a hugely accomplished illustrator. Then you may have heard of Dr. Temple Grandin. She's one of the most famous people with autism. She's written books. She has a PhD in animal husbandry. But then we have 20% of people with autism have what we call profound autism. They have IQs below 50 and they don't talk. So here you have somebody going off and getting a PhD or a law degree, and then you have somebody who doesn't talk and they have the same exact diagnostic label. So what do you make of that? How do you do research? How do you help people? And in our center, our mission is to understand each child's unique clinical and biological profile so that every child can reach their full potential, because we understand who they are and we're discovering biomarkers of different subtypes and different profiles, so that it's not just a big sort of conglomeration hodgepodge. And you probably have heard that the rates of autism have been going up. The Centers for Disease Control is the body that has been tasked with figuring out prevalence in the United States by having lots of different locations around the country determine the number of people with autism, and they average them together. And when they first started in 2020, the prevalence was only 0.6, so less than 0.5% of kids had autism. Then it went up to 1 in 50, 1 in 110, 1 in 88, and so on and so forth. And today, the rates of autism are 1 in 31, which is over 3% of kids in America. And in San Diego, the prevalence is 4%. That's an enormous increase. And people always ask, okay, what's going on? There are lots of reasons. And, improved awareness, right? We're now much more aware about autism. There's reduced stigma. I mean, Elon Musk and Bill Gates, everybody's coming out a broader definition so more people can be included. Prematurity is a risk factor for autism, and more premature babies are living, so therefore you're going to have more autism. There's universal screening programs like my Get Set Early program, where now we're just catching more kids. Also, parents are having kids older, and being advanced maternal and paternal age are risk factors for autism. Children back in the day when the CDC first started doing that, nobody understood autism so much. So in the books, they would just count it as having DD, developmental delay, not really autism. But now that we really understand autism, kids who were previously counted as DD are now check ASD. There's an increase in these autism advocacy groups. Everybody's coming out, everybody's getting together. Insurance now covers autism treatment. So if you want to get your free treatment, you need to get that diagnosis, and parents are going out getting that diagnosis. Because of that, there's now an explosion of autism treatment agencies. But probably the biggest reason that no one actually talks about, I think I'm the only one who talks about it, is what I call the digital revolution. Back in the olden days, when the CDC first started trying to count the cases of autism, you literally had data abstractors grabbing paper from, files and just going through it. Now, okay, you just press a button and you get all the data sources downloaded onto your computer, and then you can count the cases. So this is a huge reason that is not spoken about. But those are only 11 top 10 reasons that I came up with. There's probably more. So, and the Centers for Disease Control that puts out these prevalence, they also, as I mentioned, 20% have profound autism, which is IQ below 50, but 40% have intellectual disability, which is an IQ below 70. And if you go even just up to 85, which if you've met somebody with an IQ of 85, they still need some help. You're talking like 65, 70%. So a large percentage of people with autism need support. As I said, 65% of IQs below 85. But what's interesting is despite all this research and awareness, the average age of diagnosis has remained around 4 years since the CDC has been reporting this. And this is a problem because, as we know, plasticity is really, really high during early development and it goes down with age. So 4 is just too late. And it's more shocking when you think about it because autism begins during prenatal life. Imagine you diagnose somebody with cancer or something like that, and you're like, oh, let's just wait a few years. Or you knew that it was there, but you just are waiting. Autism begins in the womb. And we know this from lots of lines of research, but just I think the most compelling research is a postmortem research that was been spearheaded by Eric Crochet in our lab, where basically he got brain tissue from people with autism who've passed away, as well as people who were neurotypical, gave them to stereologists who were blind to the diagnostic group, and the stereologists just went around counting the number of cells under a microscope and gave us back the results. And then we told them the diagnostic group and they revealed the results and gave them back to us. And what you can see here on the x-axis here is we have the autism group and the control group, and this is the number of neurons in dorsolateral prefrontal cortex in billions. And you can see that in the autism group, it's over 1.5 billion neurons. And in the control group, it's less than a billion. That's an enormous difference. And we know that brain cells are generated prenatally, right? That's when all your brain cells are made for the most part, aside from, people fight about whether you can create new neurons in the dentate gyrus when you're older. But for the most part, right, all your brain cells are made before you're born. So if somebody with autism has all these extra brain cells, that means something happened during prenatal life. There's also research from brain cortical organoids that Eric Crochet and colleagues and Alison Moutry and Bonnie Toulouja Mel Campbell have done, basically taking, we create a blood sample, we take a blood sample and they create iPSCs, and then we grow out and create BCOs. And these brain cortical organoids, when you measure their volume at 2 months in age, you can see the ASDs in the pink box and the TD means typical development is in the green box. You can see that the size of these organoids are much larger. So this is all going on during pregnancy, during prenatal life. And if all this is going on during prenatal life, if you took an MRI scan of a baby at, let's say, at 1 or 2, which we also have done, you're going to find increased volumes. And again, Eric Crochet and colleagues was one of the first people to discover this. He published a paper called Unusual Brain Growth Patterns in Early Life in Patients with Autistic Disorder. And he found 12% increase in gray matter just on an MRI scan, which is a lot. And it's been replicated by over 44 MRI studies. Somebody just recently published a meta-analysis with over 3,000 subjects. So early brain overgrowth is a big phenomenon in autism. And, that's another lecture, but basically kids have too many brain cells. And so we believe that the neuron sizes, even though these BCOs are larger, if you look at the individual cells, they're a little bit smaller. And so there's too many brain cells, more connections, which of course is going to be a little noisy, inefficient. And so that's why people with autism, we believe, have a lot of challenges. So more isn't always better. I mean, but it might be that's why some people with autism actually they've leveraged their enormous number of brain cells and they're doing amazing. They can do math problems and they have perfect pitch and they do amazing things because maybe for them there wasn't as much noise. They just got the like the power of having all these extra brain cells. Nobody really knows, but it's really interesting. And if we look at this slide from Connell, it just shows nasal stain of frontal cortex neurons. And we look at a newborn, you can see cell bodies are tiny and they're still small at 1 month, but around 6 months, these brain cells are starting to talk to each other. And by 2 years, there's a lot of local circuitry in frontal cortex. But the average age of diagnosis, as I said, is between 3 and 4 years. And so what we're trying to do in our center is to develop biomarkers so that we can initiate identification and get kids in intervention before all this circuitry is happening. Because once it's formed, it's really hard to go back and sort of correct it, right? Isn't it way easier to just identify as young as possible so that you can help shape really effective connections as the child's growing up rather than like after the fact? And so to do so, because the mean age of autism diagnosis was 3 or 4 in America, when I first started doing this, I was like, well, I need to figure out a way to get kids earlier. So I developed the Get Set Early model, where I created a network of over 200 pediatricians in San Diego. I personally went and talked to all of them and said, hey, let's have parents fill out a parent report screen, and any baby that fails that, come have them send them to our center as young as 12 months in age, and then we'll do a brain scan, take a blood sample, do eye tracking, do clinical phenotype work on them. And that is how our lab finds kids with autism as young as 12 months in age. And it's great because it stands in contrast to some other people have tried to study autism younger using what's called the baby sibling approach, where if a mom has a child with autism at home, there's a 20% chance the second baby will also have autism, and they say, hey, please come in. But you can see that if that's your sample, you're only studying multiplex, autism as it occurs in multiplex families, and the genetics might be different. Everything might be a little different. So we're one of the only labs in the country where we can find autism at 12 months in the general population using this approach. And I call it Get Set Early because S stands for screen, E stands for evaluate, T stands for treat. And this is a screening tool that the parents fill out at the Well Baby Checkup. And when you do it this way, you get around 40% have some delay, not autism, using this particular screening tool, and 35% have autism. And then we do all these workups. And then 25% are false positives, meaning the baby failed the screen, but they're normal. And of course, you need normal controls for your research. So this is how we do it. And we have 243 pediatricians all throughout San Diego participating. We've screened over 150,000 babies in San Diego and over 40,000 more in Phoenix. So we know, though, that autism begins in the womb. It's a biological condition. There's genetic differences and brain structure differences and brain function differences. And yet, despite the fact that there are probably more biological differences in autism than almost any condition you could study, the diagnosis of autism is subjective. It's just based on a psychologist's opinion if somebody follows these particular criteria profiles. Leo Kanner first identified autism in 1943, but didn't get into the DSM until 1980. And then there's a woman named Kathy Lord who tried to make it a little bit more objective and quantitative. She said, okay, let's just have a test. It's called the ADOS, the Autism Diagnostic Observation Schedule, where a psychologist or a psychiatrist performs certain presses, and then a baby gets a quantity count based on how they perform on these presses, or a toddler or an older individual, and then we can sort of add up the points. So that was a step in the right direction. And then the most recent DSM has come out in 2013, where they did away with Asperger's that used to be part of the spectrum and PDNOS. Now it's just autism spectrum disorder. And as I said in the beginning of the lecture, everybody's in there now, and they threw away the requirement to have communication delays. But what is in here for all of these, doesn't matter if it's 1943 or two, it doesn't matter. Autism is a disorder of social attention. Individuals may not pay attention to your face. They may not make eye contact. They may not attend to speech. Some kids, you might call their name and they may not, pay attention. They might not respond, or they might be having a conversation and you're thinking they're not listening to the speech. They may not pay attention to other people. So maybe, they might make good eye contact, but they're not going to watch other little kids playing. They may not follow the gestures of others. So let's say you point to something, like if I pointed, you might be like, where is Karen pointing? And you might, be really interested to look. And so, yeah, so these are all instances of social attention. But wouldn't it be great if we had something that was, given that autism is biological, objective and quantitative with high test reliability, some sort of more biological marker that we could use to discover who has autism, figure out what their profile is. And so that's why I spend a lot of my research time doing eye tracking. Eye tracking is great because it can reduce the age of first diagnosis, because you can just do it. You can do eye tracking with a baby. You can reveal distinct biological subgroups by clustering kids together based on their eye tracking profiles. You can also use eye tracking as a prognostic indicator. So meaning that however a baby does on their eye tracking test at 12 or 24 months actually will predict what they're going to be like at school age. You can maybe guide treatment protocols for kids, like, oh, you have this profile and you have that profile, therefore I think your treatment should be this. And it could also potentially be a marker of treatment response for like randomized control trials. You can have kids do an eye tracking panel, test some intervention, whether it's behavioral or pharmaceutical, and then do the eye tracking again. It's objective, quantitative, and it doesn't rely on clinical judgment. And it measures social attention, which again is the key and feature of autism. So if you have never done eye tracking research, you don't know, I mean, how the eye tracking necessarily works, but it focuses on this approach called PCCR. It's a pupil center corneal reflection technique where a baby sits down on their mother's lap or anybody, and there's like an eye tracking machine that's just like right on the bottom, and there's infrared light that comes out. It's naked to the human eye, but it bounces off of the baby's cornea. So here we have a picture of the eye, and you have the pupil in the center, and then you have the cornea, which covers the eye. And there's light that is reflected off the cornea and read back into the machine once there's a reflection. So here we go. You have the eye tracking machine here on the bottom, and there's a light that's emitted, bounces off the baby's eye. And so it's near infrared light beams illuminate the pupil and creates reflection patterns on the cornea. Then there's also cameras in the eye tracking machine that takes pictures of the movement of the eyes. But it's also the cameras also help us calculate the gaze point, which is the vector between the center of the pupil and the reflection on the cornea. So we get like a little close up. I don't know if you can kind of see it, but obviously here's the pupil and here's the corneal reflection. And then here, this vector tells us where the baby is looking, whatever that distance is. And we can capitalize on the fact that babies love to look at faces. So we can use eye tracking to kind of capitalize on this fact. So there's a study by Gorin that was published in 1975, replicated by Johnson in 1991, where a mom just gives birth and a researcher, they got IRB approval for this to go in, and there's a paddle here. And the researcher just went like this in front of the baby and just recorded how much time the baby looks at a paddle that has a little face on it, a scrambled face or nothing, just a completely blank paddle. And not surprisingly, the babies look at this face more than they look at other things. So people have argued that we're genetically programmed to attend to human faces from the second after birth. And it makes sense, right? Because you need a grown up to take care of you and teach you how to talk and all this stuff, but it really is genetically programmed, most likely. And so knowing this, I developed my first eye tracking test, which is called the geometric preference test for autism. It's a 1 minute video, and it has on one side little kids, dancing around. And again, there's a biological imperative for little to look at human faces. But on the other side, there are these mathematical repetitive shapes. And some kids with autism, they are really good at math. They actually have, they might spin a car wheel or look at a fan. Like there's a preference for repetition. So I thought maybe some kids with autism might prefer to look at these repetitive shapes rather than looking at the other little kids. And I want to isolate visual attention so there's no sound. And again, it taps into kids who may have enhanced visuospatial abilities and local processing, or an interest in repetition or superior math skills. And so then all we do is we just measure the number of eye tracking or eye gaze fixations that fall into this social side, the side with the little kids, as well as the number of fixations and the amount of time the kid spends looking at the geometric side. And here's the data for a typical 14-month-old. This isn't just one example. The red dot is the actual eye gaze data. So that's the baby when they're watching this video and they don't see the red dot, but that is their actual data. So this is a 14-month-old neurotypical kid. And you can see when he's watching this video what he likes to look at. And if the dot enlarges, that means the XY coordinate is not changing. So the fixation location is the same across time. So that means they're staring basically. And now here's a toddler who was eventually diagnosed with autism when he was 15 months old. So a doctor referred him to us. We did eye tracking with him, and this was his results. And all the psychologists that do the developmental evaluations, which is we use as ground truth for discovering these biomarkers, obviously don't know the results from the eye tracking when they're doing their evaluations. You can see he starts out looking neurotypical, but then he makes a different choice. And you might be thinking, oh, this is just one example, but it's not actually, it turns out. I'm going to show you the raw data. So here's the raw data from one of the largest it is the largest eye tracking study published to date in the field of autism from a single site with 1,863 toddlers. Here, because it's a preferential looking paradigm, meaning that the baby can either look at the social side or the non-social side, every dot is a child. And here on the x-axis, we have 725 children with autism, 103 that had some features, but not enough to meet diagnostic criteria. Global developmental delay, language delay, other. TD means typical development and typically developing siblings of a proband with ASD. So we have all these different diagnostic groups. And here, you can see that this baby looked at, this is percent fixation in dynamic geometric images. 95% of the time, that means that by default, they were looking at the social side 5%. So you can know exactly how every baby performed. And if you kind of create this magic line at around 69% fixation, what do you notice? Who are the people that are kind of focusing on the geometric shapes more than anybody else? We had 125 toddlers look at the geometric shapes more than 69% of the time. So there's 125 true positives if I create that as a diagnostic threshold. But out of the 1,138 kids in the study who did not have autism, there are only 29 that performed that way. And we didn't necessarily do longitudinal diagnostic evaluations on everyone. So I'd be willing to bet that a couple of these actually will end up with autism. But if you crunch the numbers in terms of trying to identify sensitivity, specificity, positive, negative predictive value, you can see that we're only capturing 17% of the spectrum, but that's still the largest biomarker test to date. People who do genetics research, let's say that, people with autism may have a SHANK3 mutation or a P10 mutation or any of the single genes. It's literally like 1% of the ASD population. So there really are no biomarker tests for autism. 17% is actually quite high. And specificity is very high, which is the true negative rate. So the percentage of kids that don't have autism that pass the test. So that's 98%. So that's really good. And we looked at it across ages, like is it better at different ages than others? And there's a little fluctuation, but it largely performs the same. We looked across race and ethnicity. Is it, white kids or black kids, whoever, everybody's doing about the same. But again, this whole idea of sensitivity of 17%, while the best in the field, is still not really quite good enough. So what we wanted to try to do is understand who are the kids that were the 17% that failed it. So we call them geo-responders. So this is those kids who really like the geometric shapes. Then we had the middle kids, and then we had the social responders. These kids actually perform just as well as anybody else. So that's the good thing about eye tracking. It shows the entire spectrum and the heterogeneity of clinical profiles. And you can find kids who do great and kids who don't do quite so great. And then we also, we're trying to figure out what's going on. So we looked at saccade profiles, basically the number of time they moved their eyes when they were looking at various stimuli. And the kids who like the geometric shapes are here shown in red. And this is the number of saccades per second when they're looking at the geometric images or when they're looking at the social images. So we can plot both axes at the same time. And you can see the kids who are the geometric responders were a little bit unusual. They had way more saccades when they were looking at social images for the few seconds that they did. It was like they just didn't know what to look at. They didn't know how to look at it. But when they were looking at the geometric side, the side that they really did like, you can see they actually had fewer saccades. You remember the kids were staring. So they're like, okay, I'm focusing on this. So in addition to showing overall differences in the amount of time they're doing things, they're actually the way their eyes are scanning things is also different. And then the kids that were kind of in the middle were sort of in the middle. And then we looked for relationships with clinical phenotype. So we basically wanted to see that test that I talked with you about, the Autism Diagnostic Observation Schedule that clinicians use to diagnose autism. On that test, a higher score means more severe autistic or more severely on the spectrum. And so you can see this kind of graded relationship where the kids who like the geometric shapes, they had worse scores, the highest scores on that ADOS test. But now we have the Mullen scales of early learning, which is like a baby IQ. And here, obviously, average is 100, standard deviation is 15. So lower scores are worse. And you can see the kids that like geometric shapes had a lower IQ, lower adaptive. So eye tracking actually has some nice kind of fidelity with the clinical world and how the child is doing. But again, despite the promise of all this, sensitivity of 17% really wasn't good enough. So I thought, all right, well, if I could capture 17% of the spectrum with one eye tracking test, if I developed lots of eye tracking tests and I strung them all together, then maybe I could get to 50, 60, I'm trying to get to 100%. So I developed an entire battery. So we have the GeoPref test that I just showed you, 6 tests. And so basically these first 3 tests shown in blue are what I call visual social attention tests. It's just more like, what does a kid visually want to look at? Then we have one test that tries to find a different subtype of autism. So some kids with autism might visually attend pretty well, but they have a hard time shifting their attention around in fast conversations, or if things are going on, people are pointing, other stuff's happening, they don't shift quickly enough. So that's like a different subtype. It's more of a shifting attention subtype. And so we developed a gaze shifting paradigm where an actress points basically, and we use eye tracking to see if the baby is following where she's pointing. Then we have Some kids with autism, as I said in the beginning, may not respond when you call their name, or their attention to human speech isn't as strong. And so, because eye tracking lends itself so easily to visual, you might be thinking, how are you going to use eye tracking to figure out what kids want to listen to? So I decided, hey, let's use gaze contingent eye tracking technology, which is basically where you use eye tracking and you program it so that where the baby looks on the computer monitor triggers a sound file. So like you look here, you're going to hear, let's say, for example, a mother talking, a mothery speech, or you look there, you're going to hear a car honking or whatever. And so that's how we're able to figure out auditory attention in autism. And so we have two of those tests. And then we basically pool everybody's score together to get an overall autism score from 0 to 100. With if you get closer to 100, then the risk that you actually would meet diagnostic criteria for autism goes up. So 0 to 50 is in the no to low autism risk range, and 50 to 100 is in the high range. And this is just a quick, snip of the joint attention tool. So you guys probably know what joint attention is. It's where somebody points to something in the distance, and then a baby usually will follow the point, but then they'll look back at the mother's face. So there's like this triangle of attention between an object and another person and the toddler. And so here, like the mom's pointing, the baby's looking at it. So it's a shared focus between two people. And so we think this is a really critical part because it drives, there's lots of research showing that there's a relationship between a baby's joint attention ability and their language and their social competency. So here's a test that we're going to be publishing soon. We had 728 kids with autism, and there's different scenes where the actress you can see is like pointing at stuff here. And then we draw areas of interest, like if the actress points, is the baby looking at what she's pointing to? Is the baby looking at her face? Are they going back and forth? And so this just shows you really quickly. It's going to be hard to see, but basically this is the test that the baby is seeing right here. And then the camera's going to pan out to, there's a gaze replay that the technician sees. And you can see that when the actress, let's say, points at whatever the box, you can see the red dot goes to the box. Like you can literally see the baby doing what she says to do. I don't know if you can, we'll see if it works. There's sound, but the sound's not showing up. It was quick, but did you see he did it, right? So we're doing stuff like that. And then, we want to see how many kids with autism are actually doing that. And so here's just some pie charts. And this shows the number of joint attention cycles that the baby's engaged in. If you have autism, typical development, non-AAC delay, and features. And the lightest color means that 0 means you didn't do it at all. So for the autism group, almost half of the kids just didn't do it at all. But some kids did a little bit, which is awesome. Whereas in the TD group, only 10% of the kids didn't do it at all. Delay kids, a quarter didn't, and features kids, 12%. So clearly kids with autism are most likely to not really be able to do that kind of a thing. So it's a novel way of using eye tracking technology. And then this is, we just wanted to correlate performance on the eye tracking with somebody literally pointing in real life through the ADAS test to see if there's fidelity there, and there is. So that's really exciting. And then the last kind of eye tracking test I want to talk about a little bit is this reduced attention to speech. So we know kids with autism, sometimes they don't want to pay attention to mothery speech. They may not respond when you call their name. And we've done some brain imaging studies where we have children basically go and get a brain scan during natural sleep. We put headphones on, and then we pipe in some stories, and we check to see if there's brain activation during natural sleep. And we found that when you do that, typical toddlers do show brain activation in kind of the superior temporal gyrus area. Some kids with autism who have good language actually show it, and kids with autism who have poor language don't. So it's really kind of interesting that you can use brain imaging to see this, but you can see kids with autism have issues with speech. And so we're focusing a lot on mothery speech, because mothery speech is when you really do a lot of emotion and heightened salience when you're talking to a baby. Sometimes you just talk to your baby in regular speech, but sometimes you're like, hi, sweetie, I love you so much, right? So here's what, mothery speech sounds like.
Hi, sweetie. How are you today? You are so cute. So very, very cute. Look at that
Okay, so
smile.
so that's you get the gist. It's higher pitch, slower tempo, heightened positive affect, exaggerated speech contours, high rates of questions.
Hi.
And it stimulates joint attention, right? The reason we do that is we want the baby to pay attention to us so they can learn. It improves language acquisition. So the more moms do this kind of thing, then the better the language outcomes are for the babies. And neurotypical kids show a preference for the speech. If you gave them two audio files, the baby's always going to want to listen to the motherese over just regular speech. Like, even though my talking seems fine, they'll prefer the animated speech. But first so I wanted to use eye tracking to document that in autism to see how many kids liked that motherly speech and how many kids didn't. Is that going to be a marker? But first, I had to get the babies to understand that where they looked triggered a sound file, right? Because if I just did it, they might not quite get it. So first, we did a little bit of gaze contingent eye tracking training, where we had babies sitting down, and if they looked in one side of the screen, a dog would show up and bark, and if they looked on another side, a cat would show up. And babies just had to alternate the gaze back and forth at least 2 times in order for me to be like, okay, at least they sort of know. I mean, you can never know what a baby knows because they're not talking, but this was the training that we did ahead of time. And so this little girl, I think she likes the cat better. So then she got to move on to the real Motherese experiment. This was just like a little pre-trial training thing. Interesting. What is it? And so using this technology, gaze contingent, this is our Motherese eye tracking test, where a baby sits down, and if he looks on the left side of the screen, it'll pull up a sound file of a mom talking. And if he looks on the right side of the screen, it will pull up this movie file showing cars beeping on a highway and making a lot of traffic noises.
Ginger and Coco go night night in my bed. They love my blanket. They're so cute. They purr and purr. And then Ginger wakes up and jumps on my head.
And so here's a toddler with autism, how he responds in this eye tracking test.
Have you met Ginger? And so
So I see it's very similar to what we found with the GeoPref tests, but now in the auditory domain. You're getting some kids who don't want to listen to human speech. They would prefer to listen to this. And so when you look at the data, very similar in terms of what we're getting for sensitivity, specificity, positive, negative predictor value. But here we've plotted it kind of in the reverse, where having a lower number means more in indication of symptom severity. So this is the percent of time that they're fixating on the Motherese side. And you can see that if you draw this magic line at 30% fixation, you've got 67 children with autism who show really low levels of attention to speech. And then over here, there's only 4 kids that are doing that who don't have autism. So again, so this is how we're developing all these biomarkers of autism using eye tracking technology. And basically, we're just trying to pull them all together. And we have a study now with 2,422 kids where we're looking at kids who got either 1 test or 2 tests or 3 tests or all 6 of those tests to see, are we going to get that sensitivity up higher? Because I want it to be like, 70 or 80%. And yeah, we've got over 1,000 ASD kids, 670 delayed, 597 TD. We all collected this data at UCSD, licensed clinical psychologist, blind to eye tracking, did all the diagnoses so we can see what's going on. And then we also had, I bought an eye tracking system and gave all the tests to University of Seattle, Washington. I was not there. I didn't do anything at all. Just gave it to them and said, you guys also collect so we can see what's going on. So we wanted an 100% independent replication to see, does this work? Because it's pretty exciting. So this is just raw data across all the 6 tests using kind of more of that simple threshold approach that I told you. If you're above this threshold that we define, you have autism. If you're below, you probably don't have autism. And then we also, in addition to looking at the data like that simple way, we also, of course, had to do, modern machine learning stuff where you take the data set and we split it into 20%, 80%. Then we had independent holdout set and we used random forest, logistic regression, all XGBoost, Ada learning, whatever, all that stuff. And we wanted to see if machine learning could do better than just us using ROC curves, eyeballing it kind of way. And of course, machine learning has a lot of benefits because with machine learning, you put in lots of different eye tracking features, right? You're looking at number of saccades and you're looking at percent fixation and you're looking at over total looking time or amount of time just on the eyes or the amount of time on the face. So, we have literally 18 different eye tracking features across the 6 tests that we fed into machine learning versus the simple threshold approach. It's just one thing. Yeah.
Was it always just one type of video or one video per se? Because I was just curious if you swapped out the video from dancing kids to something else, included a colored child there.
Yeah. We have, if you look at the test, so we have 6 tests, and so all of them are different. So we had for the visual social attention, we had the GeoPref test. We have what we call the complex social. We had outdoor play. So I don't know if you walked in, maybe you were.
these tests are very many things you're measuring, including the saccades and
Yeah.
getting more readout.
Yeah.
But in that one test itself, if you change the video, did it change
The only thing, the only thing I changed inside the video itself is 50% of the kids got, let's say, the social side and the non-social side. I flipped it just to make sure that they did. But across the videos, it's not like the greatest racial and ethnic distribution, but we do have different, like the mom for the Motheries video is Asian. There are kids of different racial and ethnic groups playing around in the videos and stuff like that. So it's not perfect, but there is varied representation in the videos. And because we want to, yeah, you want to make sure you have large enough sample sizes to do different subtype analyses and things you want to do. I wanted all the kids to kind of get the same videos. The only thing I switched was like the side to make sure that didn't matter too much. And so here, when we're looking at this kind of simple threshold approach where I just made the line, here are the results. And then on the other graph, it's like the machine learning. This is the independent holdout test results. Because if you present results using machine learning from the training, it's always going to be like 100% diagnostic classification accuracy. So that's not very good to show that. But this is like when you took 20% of the data, after it's all been trained, you pick your final model and you put it on this 20%. What are you getting? So here, each line is a different classification metric. So you have overall accuracy, area under the curve, negative, blah, blah, blah. So I want to draw your attention to sensitivity because remember I told you I didn't like the fact that it was like low with just one test, and that's the purple line. And here you can see that this is the number of tests here on the x-axis. If a baby gets 6 tests, 5 or 6 tests, you're starting to get up to a little over 60% sensitivity. And so just spoiler alert, that's kind of the best that we're getting so far, but that's still really good. That means I can identify 60% of kids. And if you look at the positive predictive value, this is the green line with over 90% positive predictive value. Positive predictive value is what is the probability if a kid fails this test that they truly will have autism. So we're that's really exciting. Machine learning actually did slightly better than the simple threshold approach. So with 6 tests, the machine learning sensitivity is 69% versus 66% with this, and specificity is slightly better, and positive predictive value is slightly better. However, when I didn't put this on the slide, the independent the independent test site in Seattle, Washington, the simple thresh did slightly better because it's a completely different cohort and different kids. And machine learning works by training. It makes its rules and whatever based on the sample you give it. So I think the reason it didn't generalize quite as perfectly in Seattle is because their data was just a little different than ours. For whatever reason, their kids are a little different. And so, but it still worked. It's, still generalized. And so I have a grant from NIH right now to test this out in doctor's offices. Like if you're a MD and you think, oh my gosh, I'm worried this 2-year-old has autism, you can just go down the hall and do eye tracking and, kind of what's going on with that. And so here the baby takes the test, and I developed software basically where the baby takes the test and the technician presses a button and the overall autism score appears on the screen. Then the report's printed out and it tells the doctor, like on the bottom, here's the kid's autism score. And here are the simple threshold results for each of the 6 tests. So we're trying to move, this is the technology that's licensed, trying to see if we can get individuals to use that. And again, 0 to 50 is low autism diagnostic probability and 50 to 100 is high. And now I wanted to do the last portion of my talking a little bit about brain imaging. And is there any relationship between the eye tracking results we're getting and brain development? Because if you think about it, eye tracking really is a proxy for experience, right? Like if a baby's looking kind of at the wrong place or at an unusual spot on a computer screen, not looking at social images, you can bet that that same baby is probably not looking at social stuff when they're outside in the real world. And obviously experience-dependent mechanisms play a huge role in how the brain develops. What you do, what you see, what you listen to, right, feeds how your brain develops. So we want to see if there's any fidelity in what, brain imaging can tell us. So as I mentioned earlier, in our center, we do sleep fMRI protocols where we tell the mom, do not let your baby get a nap, and we bring them in, we put them down the scanner, we put headphones on, and then we pipe in little stories like Mem Fox, it's time for bed, little sheep, the cows are out and fast asleep. And believe it or not, you're getting a lot of brain activation. So for anybody who's got like little kids at home, be careful what you say while they're sleeping because they literally are processing it for sure and for true. Here we are with the data. Here we have a sample of 31 non-ASD kids, mainly typical. We had a few delays in here. And we wanted to look at brain response to mothery speech. So we used almost identical mothery speech to what we did for eye tracking. And we had different types where it was more mild social affective speech, moderate, and really highly mothery speech. And you can see here, this is like the superior temporal gyrus region where you're expecting a lot of speech activation. And we are getting that. And then in kids with autism, we had 40 kids with autism, and you're seeing that you're not getting as much activation in this temporal cortex region in response to any of these three types of speech. And so we talked earlier when I started this presentation, the hard thing about being an autism researcher is there's massive heterogeneity. And so, what if some kids had high levels of brain activation, other kids had low levels, and I'm averaging to the other and you're getting nothing. And so that's why we're starting to move towards subtyping analyses. And so we're using this approach called similarity network fusion, SNF, where it basically, you can feed it any type of data you want. So in our case, we put in clinical data like the kids' IQ scores. We gave it eye tracking data, like how do they perform on the GeoPref test and the Motherese test. And then also fMRI data, is their percent activation high or low? And then it creates these like what are called similarity matrices where it lines kids up based on how similar they are. And then they create these big networks and then they do some kind of cluster analysis to say, okay, here's different subtypes that you're getting out of your data set. And we have a paper that was in Nature Human Behavior where Yao Chang, which was a postdoc in our lab at the time, she ran all that data in through SNF and it popped out 4 different clusters. And we know who's autistic and who's not autistic. So here we have them labeled as green, where the normal controls with just a few mild delays, and the ASDs are red. So we have 4 clusters. Normal controls are largely in clusters 1 and 2, and the ASDs are largely in clusters 3 and 4. And if we just say, okay, how well did SNF do? Let's look at the percent signal change in the brain for these 4 clusters. We have cluster 1, which are the normal controls. You can see they had the highest percent signal change in response to speech. And then if you go down here, cluster 4, those are the most profoundly impaired. And that was the ASD cases. Okay, so cluster 4 is having the lowest activation in the brain. And then we want to just look at like, okay, how do they perform with eye tracking? Cluster 4 had the lowest levels of attention to Motherese speech relative to other groups. So there is this relationship between brain activation and eye tracking and subtypes in autism. And then we wanted to just be a little bit more direct about it and just do Pearson's correlations. Like what was the percent fixation on Motherese speech? And then what is the correlation? And so then here, this is for the GeoPref test, and we color coded the kids based on if they did really well in eye tracking, they're coded in green. If they were mixed, like, middle response with eye tracking to the GeoPref test, they're purple. And profound basically are the kids that were the geo responders that love to look at the geometric shapes. And this is percent fixation on social, and here's percent signal change. And you can see that it was, a lot, really a lowest brain activation in these profound autism cases. And you get a similar finding with this other eye tracking test. And then we finally wanted to say, okay, what about kids who got like all of these eye tracking tests combined, not just one test, kind of like looking at the overall performance. And, these are small number of kids who got all 5 eye tracking tests and also got brain imaging. But you can see the profound kids are here in this graph in blue, and they have the lowest levels of percent signal change. So we are seeing some nice trends here. This was significant, and this was more of just trend, but it is really interesting. So the idea being that in the future, when somebody takes an eye tracking test, you can be like, okay, this shows that you do have autism, but also it's also kind of a wake-up call for intervention that there's not a lot of brain activation in the superior temporal gyrus region or in frontal cortex where you'd expect. At some point, it's too expensive to necessarily give everybody brain imaging. But if this is a proof of principle, if we can keep showing over and over again that there is a relationship between eye tracking and brain response, then we can maybe also do interventions pre and post-seeing if you change brain response. And then one day maybe you don't even have to do the brain imaging. Just know that it is suggesting that there's probably not as much brain activation where you want it to do. And the last thing is, because we know about all this heterogeneity, I basically am trying to develop interventions based on subtypes. So here we have a sample of 227 toddlers with autism, 173 with autism and kids who are not autistic. But here in the ASD population, the kids with autism who showed really, really low, like across every single eye tracking test that we gave them, which was here on the x-axis, there was around 11% of kids just showed very low performance. So that was 50% fixation, less than 50% in all social images across all tests. Then we had 25% of kids with autism who took 5 eye tracking tests, did really well in all of them. So some kids with autism actually do fantastic. I would propose you want a different intervention for them. And 66% of kids with autism were mixed. Some tests they did well, some tests they didn't. So this is just us trying to use eye tracking to figure out like how can we develop better interventions. And this does scale with symptom severity. So this is kind of new work that we're moving into, and we have a grant to try to say, okay, before a child goes off for intervention, let's just do some eye tracking and see who they are. Are they super high social attenders? If they are, maybe we don't need to spend all this money on this intervention for them because their brain's going to be fed pretty normally throughout life. But if they're these low social attenders, let's give them a massive amount of intervention because we know that correlates with low IQ, correlates with low brain activation. And if they're a mixed case, maybe it's sort of in the middle. And so basically, in conclusion, we know that a multimodal eye tracking test battery is better than a single. I think that when you're analyzing your eye tracking data, I like the fact that we developed them all using a simple threshold approach first. So anybody using it can really look at it and interpret it, at least at some level. I think it has greater transparency, but overall machine learning does have better performance. Also, eye tracking can capture social attention heterogeneity with the autism score. And then I think overall it just has a lot of utility for the future of autism, what we can do and what we can understand. So thank you.
Questions from the in-person audience first. Dr. Kenmore.
Thank you.
Thank you.
It's really interesting. I have a lot of questions.
Okay.
Like,
Sure.
turn it into two. One, I'm just curious because, like, I guess as a non-expert, I would have thought if you don't have a preference for social attention, there would have been kind of like an even split between the two videos. But it almost seems like they actually don't want to have any of the social attention, and it's preferred towards the.
Yeah, it's almost like, yeah,
And, like, is there a rationale? Like, is there a mechanistic reason for that?
I mean, part of it is that, at least in the auditory domain, there's a lot more complexity going on because you have to look at the facial expression and listen to the speech. And that's just more challenging to do. So you just like, okay, I don't want to necessarily have to deal with that because it's challenging. Some people have tried to figure out whether or not it's literally like a just complete lack of interest. And that's kind of controversial. I think most people it's not that, it's more of challenges in processing social information is just more challenging than non-social information. And so they know that there might, there might be this extreme phenotype that really just, have there's a complete lack of interest in social, but I think it's more has to do with more processing challenges.
Yeah. And then the second question, not related to that, but I guess like with the phenotyping work that you're doing, or the subgrouping that you're doing, is that kind of what you imagine as the future for the DSM, where people, instead of how they have it now, they actually get it classified
Yes.
phenotype, and then the resources are allocated?
Yeah, let's see if I can find it.
We repeat the question.
Yeah, so basically the question was, do I see like the future of autism is really kind of moving towards subtyping as maybe the DSM will kind of do that. I have in one of my previous lectures, I made some slides about that really quickly. I think it was in, what was it? See if I can quickly find it because it's kind of cool. It was something I gave, where was it? It says CD. Oh, I see. It was this one. It was this lecture where the New York Times actually, and I'll share my screen in a second once I find the appropriate slide. The New York Times actually, in response to this whole debate about heterogeneity and autism, let me share my screen. So as I mentioned earlier, you have kids with autism who have really high IQs, greater than 85. You got 36% of kids have that. 40% of kids have IQs less than 70, and 24% have 71 to 85. And so in light of all of this, heterogeneity, everybody's thinking like, what are we going to do? And as I said, there's this thing called profound autism where you have 24-hour care, IQ less than 50, so on and so forth. People estimate different sample sizes for that, but 26% or so, or 20%. Anyway, the New York Times said, should the autism spectrum be split apart? Which is your question. Oh, you saw that fast. Okay. So you're very nice giving the credit to the New York Times. And I think that when they do, the DSM-6 is going to be coming up, and I think they very well might do that. Or right now they have kind of specifiers that go with autism. So you're a level 1, 2, or 3, and it depends on the amount of support that you need. So they might, rather than having it be numbers, they might change it at least to say mild, moderate, severe, whatever, as the specifier, so that it's a little bit clearer. Because no one even remembers 1, 2, or 3. Yeah.
do you think the word precision medicine is really yet there for management of autism? Because if you would manage the last pie chart that you showed very nicely, let's say there's a group with IQ this, and one is super. What possibly precision medicine management means where we don't even understand yet what the issue is?
That's a really good point. So the question is like, how, is there even precision medicine in autism at this point when you have such heterogeneity? Some kids have IQs below 50, some are 110, 20, 120. And no, we're not there. And that's kind of what we're fighting for at our center is like, we are saying we have to do subtyping for everything that we're doing. And we have to really sort of understand each of these different unique subtypes if we want to move towards precision medicine. So for me, I can do that with eye tracking really easily in the sense that I can give kids a whole big test battery. I can say, okay, this group of kids is more similar here, and this group is more like here, and this group is like this. And then I can try to develop interventions that are at least tailored to what I perceive their social attention interests are and profiles are as a step in that direction. Of course, one day, people, of course, I'm sure want to maybe do some biotherapeutics or something like that. But that also dovetails. That's why in our center we do, I didn't show the work, but we're doing some gene expression work, also BCO, brain cortical organoid stuff, because, some kids are really, really, on average, BCOs grow really large, but, there's heterogeneity in everything from the biological level to the clinical level. And so our job is to kind of merge all that data together to see if we can really understand what these subtypes are. Maybe there's slightly different causes, or maybe it's a unique common cause, but it's degree, which actually is what I think is going on. So autism has a common cause, probably increased number of cell cycles in the proliferation stage. And then depending on how much you have, if you've got too much of that, you're going to be really big brain, really big BCO, too many noisy connections, severe eye tracking, poor brain response and fMRI, and so on and so forth. But someone else maybe it's just a little bit of that, and that's why they have a better outcome. But we're not really sure. We're so not there yet with precision medicine, but that's what we think the right thing is to do and to try. Yeah, sure.
Is there like a very, I guess, well distributed map, I guess, of like patients who are either profoundly autistic or like less profound, like, or is it like a, there's a peak here and there's a peak here?
So
is
you're
it a normal distribution or is it
Bimodal or
yeah, that's the
Bell's?
better way.
Yeah, so if you look at the eye tracking data, if you remember, I can pull it up, but you probably remember it's continuous. So there isn't like tail ends of like severe and doing super well and then a cluster in the middle. It really is like there's a dot in every location on the map as it relates to pretty much all the types of, research, like metrics that we use, whether it's brain imaging or eye tracking or clinical phenotype or whatever. So it's really not like these severe clusters that are discrete. It's much more continuous. Yeah, and the distribution is, like that.
Dr. Pierce, from our online audience, one person asks, are we assuming that all the children have been screened for normal vision prior to eye tracking?
That is an excellent question. Yes, occasionally. So one of the questions we asked in the screening is, we personally did not screen for vision, but we asked them if they've had vision screening at their doctor's office, and they often say, yes, I have. Some kids, have a lazy eye, or they may have some vision problems. And so we do exclude them from the data analysis. So around 10% of kids are excluded because of most of the reason is because they're fussy. There's a very small percentage that are excluded for eye problems. But occasionally, yeah, they have a lazy eye, usually is the biggest problem.
But if they have a common cold that day, you probably don't do it that day.
If they have a common cold, we ask the parents just to not to bring them at all.
Okay.
So, but if they bring them, then we do it, because
Got it.
it's so expensive because we want to do a really good job, we block out 4 hours for testing. So we do comprehensive IQ testing and language, and we take a free play with a parent, and we have them fill out questionnaires, and we do a whole eye tracking test battery. And we do that across a 4-hour visit. Sometimes it's a little shorter. But then if they do brain imaging, they come back on a separate day for that. So if they come, if they're occasionally, if somebody's like massively sick, we'll send them home. But for the most part, because it's so expensive, the psychologist time that we're paying for those 4 hours is enormous. And so I want to get the data. But if somebody's real sick, we will send them home. But we usually just say, don't bring them at all if they're sick.
And then from Dr. Malhotra, can you give us a better sense of why you think autism prevalence is increasing? Is this a diagnostic awareness issue or a real change? I've heard genetic theories which don't seem likely over the last few decades.
Yeah, so I'm guessing that he may have come a few seconds late. So one of my very first slides that I showed gives my top 11 reasons why I believe that the prevalence of autism is increasing, which I'll just pop on really fast. So let me share my screen again. I think this one. So this is what I think. Improved awareness, reduced stigma. Like I mentioned in the lecture, we have Elon Musk standing up saying, I have autism, and Bill Gates and everybody. So it destigmatizes it. It's actually almost like hip to have an autism diagnosis nowadays. We have a DSM expanded definition where we're letting more kids in get this diagnosis. Prematurity is a big cause or risk factor for autism. And a long time ago, premature babies, didn't make it, but now they're living much longer. So of course that you're going to have increased risk there. We have universal screening programs. Being older as a parent is a risk factor for autism, and people are now having kids older. Children we thought were just DD, now are getting counted as ASD. There's increase in nonprofit autism advocacy groups. Insurance now covers autism treatment. And because of that, we have an explosion of treatment agencies. Like all these things gives us different sources of being able to find cases of autism to count them. But the biggest thing is this digital revolution, where in the past, people are just figuring out who has autism based on paper. And like it was arduous and it took forever and it was inaccurate. But now you can just, email a hospital, be like, oh, I'm working for the CDC. I want to count how many cases of autism you have. Bing, press a button and you digital access, way more accurate, and we're finding way more cases. So those are the top 11 reasons why I don't think, yeah, people are also looking at the environment, like maybe there's an increase in pesticide use or heavy metals or microplastics or all of those kinds of things. And who knows, maybe if you have an underlying genetic liability to be vulnerable to these kinds of toxins, maybe that could increase, the tiniest percent. But these 11 are just common sense, real factors that are the main drivers of the increase in prevalence. Is there something else? Possibly a little bit of something environmental. I'm not willing to say there's not, but I don't think it's as big of a driver as these other things.
Thank you. And then finally from Dr. Rubin, leucovorin has had some dramatic effects in children diagnosed with ASD. Have you seen any children treated with leucovorin? And does such treatment affect scoring on your visual testing?
That is a great question. Not that I'm aware of. We see babies in our center from 12 to 36 months is a predominant age. So, and we do ask, parents if they're on any special diets or they're taking any medications or whatever, and I haven't heard of that. So I would say right now, either I don't know about it or no.
Wonderful. Dr. Pierce, thank you so much for that thought-provoking and enlightening
Of
presentation.
course. Thank you so much for inviting me. I really appreciate it. Thanks,
Thank
guys.
you.
Bye, everybody.